Hello and welcome to the 8th video under cell. Let us look into cell differentiation. The process of altering the pattern of gene expression and thus becoming a cell of a particular type is called cell differentiation. The presence of chemicals or other influences start altering the decision as to which gene will be turned on or which gene will be turned off. The zygote is a totipotent cell. Its daughter cells can become any cell type. As the development proceeds, some of the cells become pluripotent, which can become many but are not all cell types. And later on, the specificity narrows down further and particular stem cells can turn into only a very limited number of cell types. For example, a few types of blood cells, but not bone or brain cells or anything else. This is why embryonic stem cells research is more promising than the adult stem cell research. Stem cell. Stem cells are the cells which has the ability to divide for indefinite periods and which can give rise to specialized cells of various tissues of the body. Two defining properties of stem cells are their ability to differentiate into other cells and their ability to self-regenerate. It can be maintained in the vitro conditions of extended periods using artificial medium. Its karyotypes remain stable even after many divisions. It can produce any type of adult cells of the organism. Stem cells are classified into four types. Unipotent, which are the cells which can make exact copies of itself indefinitely and can differentiate and produce same type of cells, for example, adult muscle stem cell. The second one is totipotent, that can become any cell type, for example, morula stage of the embryos. Then comes a pluripotent, in which almost any kind of cell except placenta can be made. For example, embryonic stem cell. And then finally comes the multipotent, which produces limited range of cell types like adult stem cell, nerve cell, blood cell, muscle cells, bone and skin cells. The sources of stem cells are embryonic stem cells and adult stem cells. Embryonic stem cells are isolated from blastocyte stage of the embryos and the pluripotent in nature, which means they are capable of developing into almost all cell types of the body. In adult stem cells, it is present in the organs but in a very little amount. They are multipotent, which means they have the ability of a single stem cell to divide into more than one cell type, but with the ability to differentiate into closely related family of cells. Embryonic stem cell. Embryonic stem cell. In this, the collection of embryos from IVF centers are done, which then are isolated of ICM, that is the inner cell mass, from the blastocyte stage. Then it is transferred the ICM to the center of the culture plate and containing feeder cells and growth medium. It can be differentiated into any cell by adding specific medium. That is it for this video. We'll come back with the next video of cell. Thank you very much.